Hello, welcome back, everybody. In this session, before the lunch break, we'll talk about Napari Zelda by Rocco. So thanks for joining us again, really. You already contributed in another part of the workshop as well. And then Napari Super Res for Super Resolution by Julian. And I understand you're collaborating on that as well.
Yes.
And then Napari Real-Time Processing from Lucien before the lunch break. So without further ado, I'd like to hand over to you, Rocco, and I'm keen to hear what you have to tell us.
Thank you very much. Thank you for the invitation, Tobias. It was nice to interact with the other participants to the workshop. So also these other previous days were very interesting, and also I learned several stuff. So it was useful for everybody. Okay, so I'll start presenting about Zelda. Now, I hope you can see the screen. I will start sharing. And now I'm in presentation mode, right? You can
Correct.
see my slide.
Mm-hmm.
Yeah. Okay. So Zelda is a plugin that I started 2 years ago, and is a plugin for doing 3D image segmentation and parent-child relation for microscopy images in Napari. So now we are at the end of the workshop, and we learned a lot of stuff. We learned a lot of coding. We learned that Napari has a very nice way of showing the result of our bioimage analysis workflow. But my question is, should we always code, or at some point we can rely on some point-and-click software? And this is the idea that is behind the plugin. So basically, looking at the surveys done among biologists, the striking result is that many of them, they face the challenge of having to analyze their data, do some sort of bioimage analysis. But the ultimate desire for many of them is to have more open source point-and-click software. And also, they look for new solution for 3D volume rendering and analysis, and the possibility of analyzing large data like histological data, very large tissue slice, and so on. On the other end, there is the fact that in some case, the lexicon used in bioimage analysis is very technical, so it's not something that is really accessible to everybody. And despite that, there have been initiatives like the Bioimage Informatic Index in order to tag software and try to achieve a way to communicate between different fields. In some cases, can still be difficult to read the documentation and understand really what a function does. So for this, I try to look at the some of the problem of bioimage analysis, and being very humble, I said, okay, these solutions are already out there. In most of the cases, if we have, for example, some images of cells and we have subcellular compartments, or, for example, vesicles, and we want to count how many of these there are for every cell. This is a problem of 2D counting or 2D segmentation. And if we want to say how many vesicles there are for every cell, we are talking about parent-child relation. And the same is true for 3D. So several types of image dataset can be analyzed in a similar way. Therefore, I started developing in 2021 a plugin that pretends to be minimalist approach to bioimage analysis. It means there is a very simple workflow that can be adopted in several cases and can bring us from the grayscale image to the segmented data. Now, this plugin is Zelda, and what I've done here is simply use the Napari viewer and enrich it with a graphical user interface that does several operations using different libraries. So in the plugin, it's possible to run a guided segmentation. So every widget that you see here at the bottom has been included to do one operation of the segmentation. So the workflow that a user has to follow is simply graphical. He has to go from the left to the right to execute the entire workflow and arrive to have results under the form of CSV file or under the form of a plot. I think is versatile because relying on the fact that Napari is a great multidimensional viewer, one can easily switch between the 2D and 3D analysis. It's possible to do the parent-child relation. That means count how many small objects are contained in a big object. It is also possible to do workflow composition. So each of these widgets is available and a user can concatenate them in a different order and can enrich the interface with new widgets and compose a new workflow. In the end, I included also a tool for doing automatic data plotting. So the data can be plotted, exported in CSV file, and the plot themselves can be saved automatically. It is, in my view, reasonably limited, so not all the data set can be analyzed with this plugin, of course. But actually, I think that every plugin should specialize in a certain type of problem. And I benchmarked the execution of this plugin against two other software that are among the most famous in the open source domain. So one is Fiji and one is a cell profiler. And I benchmarked them both in the graphical mode. It means in Fiji, one person has to do the operation manually, and in cell profiler has to use instead the test mode. And we have seen this in the previous day of the workshop. Now, the algorithm that I use for the segmentation is not my creation. I'm just capitalizing on the, let's say, on the work of giants in the field of bioimage analysis. And so what I'm doing is use some algorithm and libraries that have been written by somebody else, but I've tried to integrate them in a graphical user interface of Napari. Basically, starting from a grayscale image, it is possible with a few steps to arrive to segmentation. For example, we can apply a Gaussian blur to the image in order to homogenize the signal. And we had a very good lesson yesterday from one. We can apply we can apply a threshold to create a binary image. We can use a distance map to in order to map the pixel into a new type of image where every pixel has the intensity that correspond to the distance from that pixel to the edge of the object. We can identify local maxima, and we can run watershed. So the different steps of this workflow are run through the use of a scikit-image library, but they are done entirely graphical inside the Napari viewer. In the end, it is possible to export the measurements and produce some plot with matplotlib. Now, an example of this parent-child relation is reported here. So in this case, we have, for example, actin staining the cytoplasm of cells, and we have staining for mitochondria. And for this challenge, we want to know how many mitochondria are contained in every cell. So what we will do is first segment the parent objects, so the cell cytoplasm, and after segment the children object that are the mitochondria. And after using the function in Zelda, we will relate every parent to every children, or the opposite. We will have that for every children, we are able to obtain the labels of the corresponding parent. Now, the data can therefore be plotted with the data plotter. So it is possible to select the type of data of plot that one wants. So it could be a histogram, or it could be a scatter plot. And it's possible to feed a CSV file to the software. And the software will automatically save the plot if this option is selected. And at the same time, it will show some of the plot in the graphical user interface. So among all the properties that we measure, using the scikit image library. We can plot, for example, the mean fluorescence intensity, or we can do a scatter plot trying to plot the equivalent diameter against the mean fluorescence intensity of every identified object. Now, the benchmark that I was mentioning has been done using ImageA and Cell Profiler, like other reference software. So basically what I've done, I've tried to look at the identification that I have for the different objects using the scikit image library, using the analyzed particle in ImageA, and using the test mode in Cell Profiler. And I found that for the different category of objects, the parents, children, and after children labeled by parents label, there is a very good accordance between the segmentation that I obtained with the plugin and the other two software. I've done this in 2D, and I also have done this in 3D. And beyond doing this, I've also calculated how much time takes for a user, looking at the experience that a user can have with the graphical interface, to go from the left to the right. So from the beginning, where there is the grayscale image, to the end, where there are instead data. So I've tried to write, for example, in Cell Profiler, a similar pipeline that does all the steps, or run manually in Fiji the different step that brings from the grayscale image to having the results. And what I found is that in the graphical user interface, because I'm able to create a link from one widget to another, I can run, for example, the analysis of a data set in a faster way compared to the test mode in Cell Profiler. And however, you know, if we run Cell Profiler in batch, this is much faster. But what was my goal was develop a plugin that can be used graphically, so it was not to compete with great software like Cell Profiler. So what I've done is try to give a simpler solution for the 3D image analysis that does not require a single line of code for the user, except the installation of Napari, of course. Now, in this plugin, it's also possible to export new workflow. So all the widgets are available and can be called from a JSON file and can be combined together. So if, for example, we are able to develop a workflow that just requires 3 steps, and we want these 3 widgets appearing in the graphical user interface, we can export this workflow as a part of the JSON file, and we can redistribute to a third person. Now, it's the moment for you to try it out if you want. What you need to have is Napari installed. So there are instructions that you can follow to get Napari, or you can simply launch your Napari. And you can find the last version in the GUI of Napari, so can be installed directly under the menu plugins, installed packages. And so what I'll try now to do is try to do a small demo with you. Okay, so I have a freshly created environment where I simply have installed Napari, creating the environment with Conda or equivalent, and after pip installing Napari. And now I'm launching the software. And I'm going in the menu to install new plugins. So I'm typing here in the filter search box the name of my plugin. So is Zelda, and I click install. Now, in the meanwhile, you have in this page, in the Jupyter book, you have the description of the plugin, but there are also the links. There is the link to the presentation, to the article. There is the link to the image dataset. So there is a dataset that is 2D and 3D. And there is a link also to the installation instruction. If something goes wrong, I also provide a YAML file, so you can install you can get the same environment using this YAML file. What I'm using at the moment is this data set that is available on Zenodo. So I have basically individual slices or the entire 3D Z stack of a sample that is a tissue slice showing kidney glomeruli. And this one is the link to the presentation. Now, the plugin is installed, so I can launch it here. And you will see that there is on the right side a description of the protocol. So these are basically the steps that one could follow to have this very minimalistic approach to bioimage analysis. So in the base version, as you install the plugin, there are just few protocols. There is one that allows you to segment a single population. So if you imagine you have nuclei and you want to segment them, this will be the protocol. The second one is instead segment two population and relate, and this is for the parent-child analysis. Third one is the data plotter. So if we have previous results, we can directly plot them without having to repeat the analysis. We have a way to design a new protocol that will concatenate the different graphical widgets one with the other. And there is a way to import and export protocol. So what I'll do here is launch, for example, the segment two population and relate. Basically, in the graphical user interface that you see here, there is the same workflow on top and on the bottom, and it is meant to be applied in the following manner. So on top, we will segment the parent population, and in the bottom part, we will segment the children. And after, in the end, we will relate the parent with the children. To do so, I will use a data set that is also linked, and it represents the staining of a cell nuclei using DAPI. So you will see that this one is the stack representing the cell nuclei. I can show it in 3D. And I have another stack that represents RNA that is present in the cell. So what I want to do here is try to relate the RNA to the nuclei. In particular, I want to count how many RNA spots there are for every nuclei. So what I'll do is change the lookup table so we can see the objects in a different color. And I've also introduced one widget that allows us to rescale the data set. So for example, we can specify that the sampling we have done was with a different pixel size. So for example, we can specify the XY direction, the XY size, sorry, the pixel size, or we can specify the Z step that has been used when we were acquiring the Z stack. And this will rescale the data set, and I can do the same for the two different channels. So this will be the second one here. And I will rescale both the two stack. And now I will continue segmenting the parent population, which is the one composed by the nuclei. So I will apply a Gaussian blur. I can change the sigma of this Gaussian blur and obtain a result. This is the blurred data set. And after, I can apply threshold on this blurred data set with this option, and I will get threshold image. So as you see, I'm already having the value that works because I've included the persistence in memory in the plugin. So you will remember the old setting that we used. And of course, I've tested this demo before, and the value that I find here is correct. But for example, if I put a different threshold, of course, I'll get a slightly different result. this one compared to this one. And you will get in the name of the new layer the setting that you have used. So it should be easier to identify the right layer that you should use. Now, I will generate the distance map. This is done over a binary picture. And we'll generate this. And now in this distance map, I will find the local maxima. The function in SegImage that finds the local maxima asks you at least one image that is the distance map and the other that is the mask. And so I'm using the binary picture. And also there is a parameter that tells how many points we want to find. So this is called the minimum distance. We saw it yesterday with Juan. He explained very well this aspect. But basically, if I put a lower value here, I will find more points, more local maxima. And if I put a higher value here, I will find just few points. With this, I'm able to generate the seeds. And if the result is not satisfying, I can put a bigger distance, for example. So I will delete the layer and run it again. And I find just fewer points. Now, having this, I can use the points and the distance map to apply the watershed algorithm. So I will select the distance map, as the label says here. I will select the threshold, as indicated here by binary. And I will select the layer that corresponds to the point selection. And with this one, I'm able to segment the parent nuclei. And of course, you will notice that there might be some issue with this, but we learned yesterday the trick. Either I go back here and change the minimum distance to obtain fewer points, or directly in the layer, I can select the point layer. I can select this point here and eliminate this point. So hopefully the watershed algorithm will work better, and I will just have this cell labeled with a single number. Of course, I'll go back here, I'll delete, and I will run again the segmentation now. And now we see that the segmentation improved. So you can do this to graphically correct the results of the watershed algorithm. Again, no single line of code, just graphically do everything. Now, I will try to run the segmentation as well for the children objects. So I'm selecting the channel 01, which corresponds to the RNA. And eventually, all the other layers can be just set like invisible. And now we are setting invisible, also the distance map here. And we're just left with the original data set. Now, if I select the channel number 1 and I apply a Gaussian filter, I will be obtaining another layer that is a smoothed data set compared to the original. On this, I can apply the threshold. And I'm obtaining, for example, these dots here that you see in these blobs here that you see in white. And over this one, I can at the same time obtain the distance map. So this is the distance map over the blobs. And once I have the distance map, I can generate the local maxima. So I will select the distance map of channel number 1. I will select the threshold image of channel number 1 and use a minimum distance, which is smaller because, of course, these objects are smaller than the parent one. Now you see the point selection. And again, here you can tune this parameter in a better way. You can change it in the graphical user interface, or again, you can go in the point selection and eliminate a few points if you think they are wrong. Done this, I can use the distance map again, use the binary picture, and the second point layer. So it will be maxima channel number 1. And this will result in the segmentation of the children objects. So now I'm going back and I'm just hiding the other data set. So I'm hiding the first two. And what I'll do is show this. And what I want to show is as well the labeled parents, as you see. So the final step will be connecting, or better, relate the children objects that have individual labels with the parent objects. So I'm doing this in the last module, and after we do a break for question. So I will segment the first label object, which is the parent, and the second one is the children. And I can set the measurement over the original image. It means, let's imagine you want to look at the data from the point of view of RNA spots. You want to quantify in every RNA spot, which is the mean fluorescence intensity, all the descriptors that we have seen before, but also you want for this RNA spot to identify which is the parent cell. So we have to select here the fluorescent image of the channel number 1, which is the RNA. And we can specify a folder where we want to save the results. And with this button, we will have the measurements. Now you will see there might be some warning. So for example, for some of the objects, it is not possible to calculate some of the features. And this depends on the size of the object, up to what I understand. Some objects are very small, so it's not possible to calculate this feature. But for all the other, I will have the measurements. So I go here in the result folder, and I'm able to see that this file here generated at 329, which is right now, is the result of the analysis. So I have for every object, the label of the object, the parent cell to which they belong, the area, the volume. The volume has been calculated taking pixel size and multiplying it by the Z step that we set in the first widget here. And I have all the other parameters. So I have equivalent diameter, mean fluorescence intensity, solidity, and so on. So this was the second protocol. We segment two populations, and we are able to relate the parent with the children objects and get results. Now, I will take some question if there is any. I didn't see the chat, or I can continue and explain another protocol. That is fine. Tobias, can you just?
Hey, Rocco. Yes, there is a question by Monara in the chat. And if anyone else wants to ask a question, you can also just raise your hand and then unmute yourself.
Okay, so I'm reading here on the screen. If you want, I can read the question. So, is it possible to segment bright field image to be the parent? I would say yes, as far as you can find a method to generate a binary mask out of this bright field image. The point with the bright field image compared to fluorescence is that in fluorescence, you tend to have an object that has a certain intensity. And it's simply you set a threshold, which is a value above which you consider pixel to belong to the background. In bright field, given the technique that you use for acquiring the images, the histogram of the image is different. You might have that in the same object, you have bright and dark pixels. So setting just one value doesn't work. You have to use a small trick. You have to use some filter. It might be a variance filter or other filter that unfortunately are not implemented here in the plugin. But having done this step of processing, later on you might hope to apply threshold and get this bright field image as the one that contains the parent object. Second question, yes, you can add points as well to the segmentation, and this is great because I want to be very humble. What I've done is just connect things that they exist already, and using this amazing viewer, which is Napari. So Napari gives you the possibility to add points. We have seen this yesterday in the lesson that Juan made. So adding more points will give more seeds for the watershed segmentation. And we can apply a filter, yes, after the segmentation, but the result we will get will be slightly different. But yes, I can take layer from this. And for example, I can try to apply the same filter. And now you will see that the result of a Gaussian filter applied on labels, which doesn't really make sense for the purpose of the segmentation, is given here in the last layer here. Let's see. I will center again this, and now you should see the results. So we'll hide the other layer that are visible, the parents, and I think there is still some layer which is set as visible. So I have this hidden here. Let me see. Okay, no, this is the result. So if I visualize it in 2D, here, I'll be able to see what is the result. Let me see if I can visualize. So you see this label probably was 15, and because of the Gaussian filter, or 16, and because of the Gaussian filter, it starts to take values that include the decimals. So it doesn't really make sense, but it's not there is no limit. You can take whatever layer from here and process it with the same filter. I will look at the other question.
Yeah, there is a new question there from Alejandro. He says, is it possible to export the binary masks or the outline coordinates from the segmented cells?
The export value is not implemented in the plugin, but actually in Napari, what you can do is save the selected layer. And so this will give you some material to work on. Not filter like Gaussian, but selecting size of the spot that I would like to analyze. Oh yeah, so this is a filter by area, for example. It is not visible here in the widget, but you can call, you can create a new protocol that does include a filter by area. So simply what you will do is look at the size of this spot, as you say. And for example, if you put a certain threshold that includes this object here, but not the smaller one, it will create a new layer that just contains the bigger one. So it's not in the base, in the basic protocol here, but if you click here and you choose the option to design a new protocol, it will bring you to another part of the interface. I just want to answer the following question, and after I will do it, because of the size of the screen, I'll need to close this one or put it to float if you want to see it. Okay, yeah, thanks. And are there any other questions that I missed, Tobias?
No, I haven't seen any other one.
Okay, so,
Very good,
so I
thanks.
can, yeah, I can just show how to design a new protocol to Monara. And after I have one last point that I want to make. So I have just 2 minutes more. So what I'll do is either set to float this panel, so you can ignore it, or for example, I'll close it. I'll try to get this viewer be smaller. And now I'm showing two things. The first thing is the data plotter. So I will start with this protocol. And now what I will feed inside this is the result table that I got from the previous analysis. So will be these results, parent, children object. So I'm simply pointing it to the file, which is in the result, and selecting that I want a histogram and scatter plot, and it will save automatically the plot. And it will save it in the same result folder. So I will select as histogram the parent label, because this gives me a way to determine how many children objects there were per cell. And I can do any other plot in the scatter plot. So I can, for example, plot the maximum intensity against the extent. Extent gives us a measure of how much our objects covers the surrounding box that will contain it, or the surrounding hull that will contain it. And as I do the plot here, this plot appear in the interface. You will notice that they are pretty rudimentary compared to the newer plugin, to the other plugin. the great work that Marcelo has shown. But this was back in 2020, the easiest technology I found. So what you will have is a plot that is automatically rescaled using the data that are contained in the plot. And now you will see, for example, that there is one object that has many children, another that has fewer, a bit more. And this is a way to count for every cell how many spots you have, similarly to what we have done with cell profiling yesterday. Again, here the scatter plot is showing one parameter against the other. And because of the checkbox that we used here, those plots are saved automatically. And here you will see, in this folder, I have quite everything against everything, because these were all the tests that I've done yesterday for demoing this. And, this is just for me a good tool for obtaining a preliminary data exploration. So you can see the result. And of course, this plot might not be the best plot you have seen, but of course, if you observe a behavior, you have access to the CSV file and you can replot them better with matplotlib. And as well, I have a log file that tells you roughly the parameters that you have used in the interface to obtain the segmentation and how many objects you have found. And now I will use just last minute to show you how you can build your own protocol. I call them protocol. In reality, in image analysis, they are referred to as a workflow, is another equivalent term. But I call them protocol to do bioimage analysis. So I will select the design a new protocol. This has access to the different widgets that are contained in the code. So you don't have to do anything. You just have to use the graphical user interface. You can restart with this protocol and you can tell, for example, now I want to build a new protocol, which is called in that way, my new protocol, for example. And I want it to have 4 different steps, and I will start the design. Now, in this drop-down menu, you have access to all the widgets that are available. So you can, for example, say, okay, I want to do the image calibration. Yes, no, maybe it's not necessary. So I'll directly start with threshold, is much simpler. I want to do a Gaussian blur. No, yes. So not necessary. Then it means that I will skip. I will just include the distance map. After the distance map, what I want to do is, for example, show the seeds. And later on, imagine you have a segmentation. You can include this tool that is filtered by area, that is referred to the question that was before in the chat. Now, this my new protocol has been saved to the database. To access it, what you have to do is, unfortunately, close the software and reopen it again, because when you launch the plugin, it look at this JSON file and it loads all the protocols that are contained there. And I've started the software before this protocol existed. So I'll have to close the software here, and I'm restarting again. So I'll close here, reopening Napari. Try to answer to this. I'm launching the software, and now I see that the new protocol appears in the list because the software looked at the JSON file, what I modified, and I can add this new protocol. And now, for example, you will notice that this part here is the new widget filtered by area. So what I'll do is select the table, the results table, where I made all the measurements, the area of the particle. And here in this layer, I can feed the labeled object that I obtained before. And with this widget, I can select the minimum, maximum area that I want to use as values for creating the new layer and filter them. I will look at the last question was here. Can you have a plot that is a mean of all? Can you activate the microphone and just specify what you what is the meaning of this? I mean, of yes.
Because you showed a plot of each cell. Can I have a plot of everything together, a mean of the results?
Okay, but that one will be just one point, will be one plot with a single point. No, I didn't implement that yet. But actually, you know, if you have the CSV file here.
Yes, we can do it using another program.
Yeah, you will do the mean and the I don't know, error, standard deviation, whatever. But is a very good point for future developments, because actually we can add this feature that is the equivalent of summarized statistics in Fiji, in ImageA. In ImageA, once you have the result table, you can click on summarize, and it creates the average plus the standard deviation, if I'm correct, and is a good, you know, a good thing to introduce. So thank you for this idea.
Yes, there is a plugin in ImageA that we can do this analysis for mitochondria, for instance, but it doesn't have the result of mean because it doesn't recognize the parent cell. So you have to normalize afterwards, after counting the number of cells in the image. But in your case, we already analyzed the parent cell, so you can have just the mean at the end. I think that it can be useful for us.
Okay, thank you.
And
thank you, Matt, if you, yeah.
And for the size, when we select the minimum and maximum size, can we see the image in the binary image, which spot are we selecting, or we have to?
We can see this widget will create a new layer that just contains those objects that have the correct size. If there is 1 minute more to be as, I'll try to run it and just see if it's.
Yes, I just was wondering if we can slide the size of the minimum and maximum, if at the same time we can look at the binary image and see what we are selecting. This could be very nice.
Okay, so I'll run just the single cell, the single population protocol. That means you just have one channel, and what you want to do is segment this object. So I'm just running fast Gaussian blur. I'm doing the threshold. Threshold maybe is not the same because that set was different. Let me see. Threshold 3000 is too low. I think for this it might be 10,000. Yes, so now you see here the new layer. The distance map is obtained here.
We can't see your screen right now, Rocco.
Oh, sorry. That is my error. I was rushing too much.
But you still have time in your session, so no rush.
Yeah, okay. So let me go back here. So what I've done is load this wonderful data set that is in Napari. It's called cells. We take nuclei. Okay, we have seen it several times. I take the nuclei now, and I apply the Gaussian blur. And as a result of this, I obtain this thing. After that, I can apply a threshold on Gaussian blur. I use 10,000, and I obtain this layer here. Now, if I take the threshold image, and I get the distance map with this button, this is the final layer. And now using this distance map and the binary picture, the threshold image, if I use, for example, 20 as a minimum distance of point, is quite fair number of local maxima. As Juan said yesterday, we want to be sure 1 point per cell. We have 1 point per cell here, okay? So this will be the seeds of the segmentation. Now, if I select this, the distance map, the threshold, and the seeds, I can run the watershed. So the watershed will find the different cells, okay? And now imagine of these cells, you want to exclude this small object. So what I'll try to do is open the widget that filters by area. Area is also a measurement of size, even if the data set is in 3D, because we look at the number of pixels. I will open that widget and I'll try to exclude these small objects. So what I do is put on float mode this panel, so I have more space. I'm sorry, I'm running on a laptop, and I will try to load this my new protocol. Now, this my new protocol accepts a results table, so it means that I have to measure the size of this before I'm able to filter them. So what I'll do is I go back in the floating layer, which was the protocol for segmenting a single population. I give the original image and I give the labels. And I'm saving this in another file. It might be another folder here. So I know where it is. I measure these objects. And now when it is done, let's see, I think it's still running. I can go here in the presentation. See what is now running. Okay, this is one of the measurements that Segmentation runs. Now this is the file. Now in this file I have this column, which is the area. And for example, I know that there is this tiny object as area 11 that is not a cell, so I can try to filter it. And the same might be true for this one that is 200 or even the one that is 7,000 compared to the area that other objects have. So the other objects have this area that is 41,000 or 30,000. So reasonably, this object that is just 7,000 maybe is a very, very small object. So what I'll do is go back in the interface, and I'm selecting the layer, which is the labeled layer. So this colored one. And I can set the area, and I want the area to be, for example, now let me see. For this, I'll put 500. And this one can be whatever value. I put a very big limit, big maximum for this to consider also other data set. Now let's see if it works. And now this is the filtered layer. So as you see, it contains less cell, and it does not contain this cell that is smaller, and also this mid one, this one in the middle, but just few of them, because according to the calculation, they had a different area. So you will see the difference. This was the original. And here, if we look, for example, this small object, let me see if I can. This small object here in the filtered layer has been excluded. And I have just a doubt about this thing in the corner, why it is showing. It is showing anyway. So I'll try to double check here if there is some odd behavior in the corner. But in principle, this should be the widget that you can use to filter. Okay, so let's see. Okay, it looked like there are no other questions.
I'll take advantage of this time, but I was wondering, Rocco, if we can do analysis like that one for 2D images, but without fluorescence, for colorimetric images. For instance, we have, I don't know, but staining of glycogen in the cell, and it's a little bit hard to differentiate the staining from hematoxylin, for instance. So the analysis is not quite easy, like as in fluorescent images. So maybe this plugin could be available for this kind of analysis using hematoxylin and eosine or other colorimetric staining.
So there is one thing is that there is another software called QPath, which is is made for this type of analysis, and it's amazing. It's another amazing software. So I would say in this plugin, what we do is rely on the fact that the images are fluorescence light. It means we tend to apply a threshold to segment them. So if there is any step that you can do to transform your data set to an image that is fluorescence like, maybe you can apply, you can split the channel of an RGB picture, whatever other way to go around can bring you to the point where you can use the plugin and use the same procedure that you use for fluorescence. Of course, the ambition of this software is not big, so it is not made to analyze any sort of data set. And for the analysis of hematocylin and eosin, QPath will work very well. So I suggest you to give a try in QPath first before you try to develop a very complicated strategy to transform this image.
Okay, looks like everybody who asked the question is happy. It was really, really interesting. Thank you very much for leading us through. I think also the combination between the presentation and doing it hands-on is really great. So that, yeah, it just makes it easier to really understand how to open it yourself afterwards and how to play around with it. It's a very useful tool.
Yeah, thank you. I also invite the people in the audience to try the plugin and write me if you have any issue, any idea for improvement. So thank you very much for giving me the possibility to present it. And I now say hello to Julian.